Hello everyone. So the cell is the structural and functional unit of life, and it's basically the one that controls our day-to-day activities. So stay with me as I show you more details about the cell. So today we're going to talk about the cell. Okay, the cell. What actually is the cell? The cell is the structural and functional unit of life. That is, the cell is the one responsible for the control of our day-to-day activities. And this cell as a whole was discovered by a scientist called Robert Hooke. So Robert Hooke discovered the cell. And this cell, like I said, controls our day-to-day activities. Okay, so I'm going to use a story to explain the cell. The cell is like a company name. So let's say there is this company named the cell, and it has many workers inside it, which is the organelles. You know, I said in my last video that a collection of organelles will give rise to what? The cell. So the cell is the name of a particular company, and it has many workers in it, and those workers are called what? The organelles. So the organelles are what is actually found inside the cell. Okay, so like I said, the cell is a name of a company, and the workers inside are what? The organelles. So I'm going to give you the organelles, the function of the organelles found inside the cell. That is the function of those workers found inside the cell as a company. And now we're going to talk about the protoplasm. The protoplasm is made up of the nucleus and the cytoplasm, and that makes it nucleus plus cytoplasm will give you what? Protoplasm. And that makes the protoplasm the CEO of the cell, is the founder of the cell. You know, I said that I'm going to use a story to explain this. So the protoplasm is the CEO of the cell, and in the cell term, the protoplasm is made up of what? The nucleus and the cytoplasm. And now let's talk about the nucleus. What actually is the nucleus? The nucleus is the managing director of the cell company. The nucleus is the managing director of the cell company. So therefore, the nucleus is the control center of the cell. Did you just hear that? The nucleus is the control center of the cell. It is the managing director of the cell company. It carries hereditary materials like DNA, RNA. So therefore, making it what? The control center of the cell. Now let's move on to the next one, which is centralis. Centralis is responsible for cell division, the growth of the flagella, for movement. So what are we going to call that now? Let's say the centralis is the workers or the people there that make sure that the company grow and the company what? Move forward. Therefore, making the centralis the man responsible for what? The production of all those flagella used in movements. Okay, used for movements. So I don't know if you understand this scenario I'm giving here. The centralis is responsible for production of the cilia, the flagella used for movement, the spindle fiber, all those things used for the growth of the cell company. Okay, so let's move on to the next one. And now that brings us to the ribosome. What actually is the ribosome? The ribosome is round in shape, and it's found attached to what? The endoplasmic reticulum. I'm going to talk about that, the endoplasmic reticulum. So the ribosome is found attached to what? The endoplasmic reticulum, and is responsible for protein synthesis in the cell. So what are we going to call the ribosome in the cell company? We can call it the chef. You know, the chef are the one that cook. So because of the protein there, we can just name it the what? The chef. the chef of the cell company. So the ribosome are the chef of the cell company. So let's move on to the nucleolus. The nucleolus are responsible for synthesizing in the cell. So that's an attachment. I don't think I have any place to put that in the cell company. Let's move on to now the chloroplast. The chloroplast is made up of green pigments called what? Chlorophyll. And what exactly is this chlorophyll? This chlorophyll is responsible for the green coloration of the plant. It's as simple as that. The chlorophyll is responsible for the green coloration of the plants. And this chloroplast is responsible for what? Photosynthesis. The chloroplast is responsible for photosynthesis. Photosynthesis. How do they pronounce it again? The chloroplast is responsible for photosynthesis. And photosynthesis is the process whereby plants attract sunlight to them and produce food. I'm just giving you the most basic explanation of photosynthesis. Process whereby green plants attract sunlight and undergo some processes to produce food. So let's move on to the next one. And now that brings us to the word, the lysosome. Oh, I forgot to say something concerning the chloroplast. The chloroplast is also known as the kitchen house of the cell. Note that the chloroplast is also known as what? The kitchen house of the cell company. Okay, you can say the chloroplast is known as the kitchen house of the cell, or you can say the chloroplast is known as the kitchen house of the what? The cell company. Because you see, it actually carries out photosynthesis, and photosynthesis produce food. So the chloroplast is the kitchen house of the cell. It's as simple as that. Now let's go talk about the lysosome. The lysosome is responsible for digestion. That lysosome guy is a very clean guy. Any bone-out tissues, any waste, the lysosome will remove it. Like the lysosome doesn't take any time to just remove all those debts out of the cell. And that's why they call the lysosome the suicide bag of the cell, or you can say the suicide bag of the cell company. You know, we're using a company basic to defend this, and we're still using a cell theory to defend this. So you can call the lysosome the suicide bag of the cell because it destroys worn-out tissue. Like all the worn-out tissue that are in the cell, it will destroy it. It will carry out digestion, remove anything that is unwanted, all those waste material. The lysosome is responsible for that. Now let's go to the next one, and that brings us to the Golgi apparatus. Hey God, the Golgi apparatus. The Golgi apparatus, that guy is like a close friend to the cell. The Golgi apparatus. God, the Golgi apparatus is responsible for manufacturing, packaging and exporting. Only one guy to manufacture lysosome. It will pack and export materials out of the cell. it's like the messenger of the cell. It's like the messenger of the cell. The Golgi apparatus is like the messenger of the cell. It manufactures, pack and exports materials out of the cell. That's crazy, right? So let's talk about the endoplasmic reticulum. So this endoplasmic reticulum is one of the workers in the company that has two functions. You know, the rest has one function, or let's say more than one function, but just one branch. This endoplasmic reticulum has two branches, and is the smooth and the rough. So what exactly does the smooth endoplasmic reticulum do? The smooth endoplasmic reticulum is responsible for synthesizing what? Lipids and steroids. And the rough endoplasmic reticulum is responsible for protein synthesis. That now brings us to the ribosome, which is also what's responsible for protein synthesis. You just see that the ribosome is also related to what? The rough endoplasmic reticulum. So you just see the functions of the endoplasmic reticulum. It has two branches, which is the smooth and the rough. And now let's talk about the plasma membrane. The plasma membrane is like the bodyguard of the cell, or let's say the gate man of the cell. The plasma membrane serve as a guide, or let's say the barrier from, like, it allows things to go into the cell and it allows things to move out of the cell. Without that plasma membrane, things will not be able to go out and come in freely like that. It's like the bodyguard of the cell. So that plasma membrane is responsible for what? The passage area for things that are coming in and going out of the cell. Do you understand that? So let's move on to the next one. And that now brings us to the cell wall. You know, I said plasma membrane is the gate man of the cell company. So this cell wall is the main gate. It serves as a barrier. Like, it's the main gate. At least plasma membrane allows things to go in and go out. You understand now? But cell wall is the main gate of the cell. It protects the cell against harmful substance. It's like the security, the main security guard of the cell. Protects the cell against harmful bacteria, infections, and all those stuff. So the cell wall is the main gate of the cell. Now let's go to the mitochondria. The mitochondria is regarded as the powerhouse of the cell, or the powerhouse of the cell company. Let me tell you why I said that. The mitochondria is responsible as the mitochondria is called what? The powerhouse, because it is responsible for energy production. Do you just hear that? The mitochondria is responsible for energy production. And the mitochondria also have another function, which is cellular respiration. Cellular respiration is breathe in, out, breathe in, out. So that's cellular respiration. Okay, so mitochondria is responsible for cellular respiration and also regarded as the powerhouse of the cell. It's responsible for energy. Okay, so let's talk about the other one, which is the what? Cytoplasm. Now, the cytoplasm is like a jelly-like structure. What name are we going to give the cytoplasm? Let's call it the water supply, because it's actually responsible for all metabolic process in the cell, and it contains mainly water. So what are we going to give it? We'll give it the water supply of the cell company. Okay, that's nice. We can give it the water supply of the cell company because it controls all metabolic process and it's mainly filled with water. That's why it's like a jelly. Like, you know, jelly structure are normally very soft. Yeah, they are normally very soft. So quickly, let's move on to the differences between plants and animal cell. I'm going to show you what is present in plant cell and what is present in animal cell, and what is not present in plant cell and what is not present in animal cell. So let's go. And now, the plant and animal cell possess both of all those organelles that I listed, like the nucleus, the cytoplasm, and all that, the mitochondria, but they don't possess the lysosome, what other things do they not possess? The centralis, the chloroplast. And note this, in a plant cell, the cell vacuole is very large, and in animal cell, the cell vacuole is very little. So because I said they don't possess all those things, that doesn't mean that one of them don't have it. Like the animal cell centralis is present but absent in plant cell. Then the chloroplast in plant cell, chloroplast is present but absent in animal cell, while the lysosome in animal cell is present and absent in plant cell. So you see the difference there. Because I said they are not present, that doesn't mean that one of them don't have any of these. Okay, so that's it. Now I'm going to move on to the cell and its environment. That's what I'm going to talk about, those osmosis, the diffusion, the crenation, the plasmolysis, and all that. And now, the first one that we're going to talk about is diffusion. Diffusion is the movement of molecules from a region of higher concentration to a region of lower concentration. Did you just hear what I said? Diffusion is the movement of molecules from a region of higher concentration to a region of lower concentration. And note this, the difference in concentration between two liquid that undergo diffusion, two gases that undergo diffusion is called concentration gradient or diffusion gradient. So that's the difference between them. Okay, the similarities, the difference between them is called concentration gradient or diffusion gradient. And now I want to tell you some factors that affect diffusion. You know, I said diffusion is the movement of molecules from a region of higher concentration to a region of lower concentration. So these are the things that affect diffusion rates. Number one is temperature. Temperature affects diffusion rates. The second one is surface area. Surface area also affects diffusion rates. The third one is the concentration of that gradient. So the concentration of that gradient also affects what? The diffusion rate. So there are many other things, etc. So I just listed 3 because I want this video to be very quick. I want to summarize many things about the cell in this video. Okay, so I said temperature, surface area, concentration, and stuff like that, etc. So now we're going to move to the osmosis. The osmosis is the movement of water molecules from a region of higher concentration to a region of lower concentration. But you know, I said diffusion is the movement of molecules, but osmosis is the movement of water molecules. You see the difference there is water. Diffusion is the movement of what? Molecules, and osmosis is the movement of water molecules. And there is another thing added to the definition: through a semi-permeable membrane. Did you just hear that? Osmosis is the movement of water molecules through a semi-permeable membrane. And here is one thing about the osmosis. The osmosis has 2 parts, which is endosmosis and exosmosis. So do you know that endo means inside and exo means outside? So what does that tell you? It tells you that endosmosis is the movement of water inside the cell, and exosmosis is the what? Movement of water outside the cell. It's as simple as that. And now I want to tell you 3 things called the hypertonic, hypotonic, and isotonic solution. So what is hypertonic solution? Hypertonic solution is a solution where the surrounding concentration is very, very concentrated and toxic to the cell inside the cell inside the concentration, or to the concentration inside the cell. What do I mean by that? When you say something is very concentrated or toxic to the cell, it means that when you dip it inside that solution, it will shrink, and when it shrinks, it will lose a lot of water. It will lose a lot of water. And the hypertonic solution is when the concentration is very toxic, right? So let's move on to the hypotonic solution. The hypotonic solution is when the surrounding concentration is less concentrated. You know, I said hypertonic means when the surrounding solution is very concentrated than that of the solution of the cell. Now, hypotonic solution is where the surrounding solution is less concentrated. When I mean less concentrated, that means it's not up to the concentration inside the cell. So therefore, it will cause the cell to what? To absorb a lot of water. It will not swell. It will not swell like this. Oh my god. So it will not swell very well, but it will not burst. You know the other hypertonic, if you hear the name hyper, it's always something very hard and harsh, something very bad. It will cause that one to what? To shrink. But hypotonic will cause this one to absorb a lot of water. Okay, absorb a lot of water. Sometimes it might burst, sometimes you just absorb a lot of water there. Okay, and now let's talk about the isotonic. Iso means normal, neutral, natural. Okay, the concentration of the surrounding and the concentration of the cell are the same, so it will not cause any harm to the cell. So are you getting that? It will not cause any harm to the cell. Isotonic, neutral, natural. Iso means normal, so it will not cause any harm at all to the cell. It will just allow the cell remain what? Normal. So now let's look at what hemolysis means. Hemolysis is the bursting of red blood cell when placed in hypotonic solution. So plant cell don't normally burst like animal cell because plant cell has a turgid cell wall. The turgidity of the plant cell is very, very massive. That's why it cannot easily burst like the animal cell. The animal cell will easily burst, but the plant cell does not easily burst because the cell wall of the plant cell is very turgid. So that brings us to the crenation. The crenation is the shrinkage of the red blood cell when placed in what? Hypertonic. So I know you already get this before I have said, because I've said it before that hypertonic solution, any cell placed in hypertonic solution makes it shrink. So crenation is the shrinkage of red blood cell when placed in what? Hypertonic solution. It's as simple as that. Now let's talk about plasmolysis. Plasmolysis is the shrinkage of plant cell and the pulling away of the lining of the cytoplasm when placed in hypotonic solution. Listen carefully, when placed in what? Hypotonic solution. So when I mean the shrinkage, when something shrink, it means what? It's become very small. So it's the shrinkage of the plant cell vacuole. I'm sorry about that, I did not mention that earlier. It's the shrinkage of the plant cell vacuole when placed in hypotonic solution. So in crenation, let me go back to crenation. In crenation is also known as what? Laking. Note that crenation is also known as laking. Laking. Crenation is also known as laking. Now we're going to talk about the flaccidity. Flaccidity is when plant loses water to the environment faster than it usually is. Flaccidity is when plant loses water to the environment faster than it usually is. And in what they call it, plasmolysis. In plasmolysis, that it is caused by excessive transpiration of water. Let me explain. I said it's the shrinkage of plant vacuole when placed in hypotonic solution and the removal of lining in the cytoplasm. So let me tell you what happened. When I mean is caused by the excess transpiration of water. Transpiration is what do they call transpiration? Transpiration is like the taking up of water when water passes through the plant with full force. So because the water that is passing through the plant is very much, it now causes that plasmolysis. That's the shrinkage of the vacuole. The water will just, like, the water is very excessive. The water is very excessive. The transpiration pool of the water is very excessive in that plant cell, therefore causing plasmolysis to occur. And if plasmolysis occur, it means that the plant cell will shrink, the plant cell vacuole will shrink due to the presence of hypotonic solution. And that's why I now went to what? The flaccidity. The flaccidity is when the plant loses water than usual. That's just what flaccidity means. I hope you have been getting all these things that I'm saying. Okay, I hope you have been getting it. So let's look at turgidity. Turgidity is the swelling of the vacuole of plant cell to become what? Turgid. And do you know what turgid means? Let me describe it for you. Look at this, my cheek. Take a look at it. Right now it's very soft. It's very soft. But if I want my cheek to become turgid, maybe I might put some water in my mouth or do like this. And once I do like this, you see that it becomes what? Strong. Take a look at it. It becomes what? Stronger than normal. So that's what turgidity means in plants. The swelling of the vacuole of plant to become what? Turgid. To swell, to become what? Turgid. And if something is turgid, it means that it's what? Is stable in nature and is what? Strong. You understand that? So let's talk about the what they call it, the active transport. The active transport requires energy. Is the transport mechanism in the cell that requires what? Energy. Active transport is the transport mechanism that requires energy. And osmosis and diffusion in the cell does not require energy. That makes osmosis and diffusion a passive transport. So are you getting me? So let's look at the last one of the day quickly, which is endocytosis and exocytosis. Like I said, endocytosis is the movement of materials inside the cell, and exo is the movement of what? Materials outside the cell. It's as simple as that. Endo means what? Inside, and exo means outside. And that makes endocytosis the movement of materials inside the cell, and exocytosis the movement of materials outside the cell. And under exo and endocytosis, they are divided into two subcategories, which is the pinocytosis and the phagocytosis. The phagocytosis is the movement of solid material inside and outside the cell. You know, I'm referring to both of them now. So it can either be inside or outside the cell, but that does not even concern us right now. We're talking about the phagocytosis, which is the movement of what? Solid materials, solid food materials inside or outside the cell. And pinocytosis is the movement of what? Guess that. Liquid food materials. Yes. Liquid food materials inside the cell or outside the cell. So therefore, exocytosis is the movement of materials or outside the cell. So that brings us to the end of this class. But not really the end. I'm going to talk about cell and other thing relating to cell in my next video. But I've just given you the basics and almost all the things you need to know about the cell. Okay, so in my next class, I'm going to see you there, and I hope you love this video. So like and subscribe for more details. Bye.